With that, we move on to the next session titled Engineering Antibody Based Precision Medicines. And it's my pleasure to welcome Dr. Jagath Reddy Junutula, co founder, NUCO, board chairman, Science Gurus. Dr. Junutula, over to you.
All right. Thank you all for listening to my talk. It's my pleasure. And thanks for the organizers for inviting me for this special meeting. And let me share my presentation. Hope you are able to see my screen. All right. What I'm going to share next 30 minutes or so, something special happened in my life 10 years ago, actually. When I visited India 2011, that's my Amma with my 2 kids. So it was a beautiful life. And about a year later, what happened was I got a call from India saying she was bleeding. And we sent her to hospital, one of the best in the country. It's Apollo, and they diagnosed and they encountered she had 2 different cancers, a breast cancer and another one is a skin adenoma cancer. And they immediately did surgery and got rid of the tumors for the skin adenoma cancer, and they removed the breast, and she went, luckily it was ER positive breast cancer, so they could remove it and did the treatment for it was easy for ER breast cancer. So I knew what's going to happen because I've been working on the cancer drug discovery for past 20 years. So within few months, I was fortunate to take a sabbatical from my company, then came to India with my family, spent 2 months with my mother. She was good, healthy. Then we returned back, and 2014, early January or so, that again returned. That cancer, which was not there when we visited, but within 6 months it returned big time now. And she refused to go to hospital because she said it's no use, and she passed away, actually. So what I was trying to say is, about a year or so she lived, but that was a good thing. I made a decision to come and spend time with her, and that was really good thing. But we spent about 6 lakhs, and I think I can afford that being in US. But imagine people in India spending 6 lakhs for living a few more months. I got a call 1 year ago from a friend of mine. The patient was stage 3 lung cancer, and patient is only like 40s, and they are getting ready for the daughter's wedding. So they asked me, what can the patient do? If you go for treatment, probably he has to spend 5 to 6 lakhs. Maybe he will live for a few more months, but his family will be broken. Is that a good thing for the family or not? My advice was for him is, let the family go get married, the daughter, let the daughter settle down, let him spend peacefully at his house with his family. Let's move on. That is the story. I was struggling past 20 years. Why can't we have better drugs? Why only patients, even after spending US, this targeted therapies, $100,000 they spent, and they only live few more months, maybe a year, maybe 2. Yeah, that's great. But why can't they be cured completely? Yes, there are some patients get cured completely. But why do we have to break our financial system for this? Can we make cheaper and affordable drugs? And how do we do that? So let me tell a background story, what I was doing for 20 years to trying to figure out this targeted therapies. These are so-called engineering precision medicines. And how do we do about that? So there are many kinds of engineered precision medicines. So what I'm going to speak about one class, antibody-drug conjugates. And what I'm going to tell about how we develop these and what is the rationale you apply, how do you go about selecting the drugs that you attach, and how do you broaden the window, and how are the combination therapies coming along, and what does the future hold for. So let me step back and tell you what is antibody-drug conjugate. Antibody-drug conjugate is a three-component molecule. It's a complex molecule, a biologic, a small molecule, attached together to make an antibody-drug conjugate. In other words, if you're thinking about antibody-drug conjugates, you have to have a thorough knowledge on antibody, a large molecule, a thorough knowledge on the small molecule, a thorough knowledge in the linker that you're attaching to make it successful. And traditionally, if you're using a chemo is all over the body. Every cancer patient gets chemo. Why would they have so many side effects? Because chemo actually works on normal tissues, also kills the cancer tissues. Because of that, you have a small window. But what does the antibody-drug conjugate do? You are attaching this chemo to an antibody to specifically deliver the payload to a targeted tissue, like a missile, targeted missile. So because of that, you broaden the window, minimize the efficacy dose. Therefore, you have a very good window. That's where antibody-drug conjugates work. As you see in the right side of the cartoon, an antibody-drug conjugate binds on the tumor cell surface, gets inside the cell, and get internalized, and goes to lysosomes, where antibody gets degraded, the payload is released, and the payload actually binds to mechanism of action and then kills the cell. That simple as that. There's a lot of history. It started back in 1910 with Paul Ehrlich, who was the first person to coin this targeted therapy, so-called magic bullet. And field went on so far. We are so good. And we had first approved drug, Mylotarg, which happened back in 2000. That was withdrawn. Again, it came back in 2017. I was fortunate to be part of Kadcyla, which was approved at Genentech 2013. I was a Kadcyla R&D team. I was also fortunate to be part of Polivy, which got approved in 2021. And I was the inventor of the Polivy. And I was part of this history, actually. For 20 years, I'm really proud about my history. In fact, what is the success? Why people are excited about this class of molecules? There are past 15 years, especially, so much going on in the field. And I'm sure you all know about antibodies. The first humanized antibody, Herceptin, everyone knows, famous breast cancer drug, works beautifully for millions of patients got benefited from that. It's a first humanized antibody was used, developed by Genentech. And it's a fantastic molecule. And why humanization is very important? People very early on, they use mouse monoclonal antibodies, and they are immunogenic because you can't take mouse antibodies and put in the patients. So they have to humanize it, use human framework. That was a disruptive technology, happened back in 90s. Then later on, Mylotarg, the first ADC, showed clinical proof of efficacy in AML patients. That sort of gave a hope, hey, we can attach the payload to antibody and deliver specifically targeted tissue and make it work. The next big thing happened in 2000s, that is 2011 and 2013, Seattle Genetics at Cetris, Genentech at Kadcyla got approved for hem-onc and solid tumors. That's the biggest news actually in the field. Hey, actually this concept does work. But what is the challenge? Improving the window. As you can see, I've plotted these publications since 1960. Personally, prepared this graph. You can see how field is growing since 2000. Skyrocketed. This is where I started my work in this field. In fact, personally, I read this 5,000 papers past 5 years to see what is really going on in the field. Are we really doing good discovery science in this field? Do we really need to make any disruptive ideas? And what is that we need to make a difference? So past 20 years or so, 183 ADCs went into clinic. 102 are still active. 10 of them were approved. And again, I am part of this. Of the 2 out of 10, I'm part of that history, thankfully. I was fortunate to witness that success. And 81 of them were discontinued. 929 clinical trials happened till to date. That means your only success rate is 10%. 10 out of 90 only success. And if you ask me these 100 which are active, most likely another 10 of 100 only will be successful. Why is it 90% are failing? Are we not doing good science as a scientist? I mean, as a scientist, I really feel disappointed that 90% of the time we are not successful. Means something must be we are doing wrong. So how does this ADCs work? Typically, ADCs bind the tumor cell and kill the tumor cell very nicely. And also in that process, tumor cells have abundance of target expression. but same target could be present in the normal cell, then that results in target-dependent toxicity. And also, when the linker fails in the blood plasma, and that failed linker could result in the payload premature release, that could actually leaks into normal tissue, can kill the normal cell as well. So in other words, a fine balance between target-dependent tumor killing, a target-dependent toxicity, a target-independent toxicity. In other words, you want to enrich the target-dependent killing, minimize this target-dependent independent toxicities. That's the only way it will work. How do we do that? As I pointed out earlier, ADC is a very complex molecule. When I say complex, it's really complex because you have three components. You have to think about tumor antigen that you want to work with. You have to think about antibody that you're working with. You have to think about linker that you're working with. You have to think about cytotoxic drug or chemo payload that you want to attach it. All are equal partners here. Even one of them you mess up, you're going to mess up the molecule. Let's go quickly. How do we go about select the target that you want to work with? Very simple. You look for expression profile in the tumor, expression profile in the normal. Look for a target that are abundantly expressed in the tumor, but minimally expressed or none in the normal tissue. That's your preference. Once you identify that, you look where the target expressed on the cell surface, where the target internalizes, where the target copy number is more than 10,000 copies per cell or not. If they're not more than 10,000 copies, you do not want to work with, because ADCs will not work under 10,000 copies. Because you have to sufficient payload you have to deliver inside the cell for make this ADC work. After you do that groundwork, then you go ahead and make antibodies, and then you need to have antibody that crosses with human antigen and also crosses NHP, non-human primate, a monkey antigen, because you have to do tox studies in NHP before going to the human. And then you have to do ADC characterization in vitro, in vivo. Then you humanize the antibodies or make human antibodies directly. And then go and look at the safety, and then eventually go to the IND studies and then the clinic. So how do we go about look for the expression profile in the normal versus cancer? There's so many bioinformatics databases available. One of the best is TCGA, the cancer, the total cancer genome atlas. Millions of data points out there. And I'm just showing you one of the example here. This is GFRI, is the breast cancer target. When we look at it, we look at red versus green versus blue. Red is the expression in the cancer. Green is the expression in the normal. Blue is the expression in the disease state. As you can see, each point expression in one human individual data point, RNA expression. As you can see, this target selectively and highly expressed in the breast cancer, but minimally or no expression in other tissues. That is the first data point you would want. Then we went and looked at all that and characterized, and indeed true. We made antibodies, fantastic, they're good, FACS positive. Then we wanted to screen which antibody is right. So we took an antibody attached to the linker payload, the chemo payload that you want. Then you test it on two cell lines. One is cell line expressing the target, cell line non-expressing the target. When you test on that, you can see beautifully, there are certain group of antibodies work very nicely, certain group doesn't work. Long story short, there are many reasons why they don't work, but we can focus on the ones which work and move on with it. I have the reasons why they don't work. I just don't have time to explain today. I'll be happy to answer offline. So taking those lead antibodies that we selected, we had to do in vivo tumor xenograft studies to make sure if you model that human cancer, in this case breast cancer in the mouse, would this ADC work? Yes. Of these 5 of them we selected, 2 of them worked, 3 of them did not work. And probably there is affinity correlation, or there could be PK correlation, pharmacokinetic correlation. But luckily for our purpose, it's not lucky, actually scientifically, the 2 antibodies, beautiful affinity, worked very nicely. Their half-life is very good. The other 2 antibodies did not work well. The affinity is poorer. Half-life we did not characterize. Having said that, we moved on these 2 antibodies actually to further. So that's another way of looking at it. There is a different way of looking at the target selection is how do we select which target is right when you have too many targets? Here an example, a B cell target. We're talking about 9 targets on the B cell expressed. Which one you want to pick it? Beautiful paper published in 2009 by my Genentech colleagues. What they have done, they made antibodies for all these targets, then screened by FACS. They looked at which target internalizes. Why do you internalize? You care? because the target has to get in. When antibody binds the target, they both have to get in, so the payload can get in inside the cell. That's why internalization is very critical. And you can see CD19, CD79, CD22, beautifully internalized, the dotted structure. All others still on the cell surface. So they moved on with these two targets, and they work beautifully. And they characterize in vivo. Depending on which linker you want to use, the typical linkers are cleavable, non-cleavable. You want to use a cleavable linker when target internalized or not internalized. If target not internalized, you certainly want only cleavable linker, not non-cleavable linker. A lot of rules around that, okay? And next we move on to payload selection. We talked about antibody, we talked about the linker, we talked about the payload. So payload, there's so many different classes: cytotoxic payload, chemo payload. Again, there are payloads are femtomolar potency, there are payloads micromolar potency. What is right for you? There is no written rule, one thing is right for one indication or vice versa. You got to figure it out which payload is good for which indication. You need to scientifically prove that, because not necessarily one payload is going to work for all. And fantastic data has been done past 20 years, so much diversity, heavy stuff on two class of payload: orostatins, the tubulin binders, and methansins, another class of tubulin binders. But they pretty much cover 50% of the ADCs that went into clinic, other remaining 50%. And this is one of the fantastic story happened. Daiichi Sankyo, very proud of their decision, choosing a different class of player than anyone else. And this, they collaborated with now AstraZeneca. This program is going to phase 3 now, and it's probably one of the best ADC that I ever witnessed. And it's going to come out and blast. I mean, breast cancer patients are going to benefit huge from this ADC, actually, in my opinion. So what Genentech already have approved HER2 positive breast cancer ADC. Why this is so different? Genentech ADC has the methansin class. That was the ADC designed 20 years ago. And Daiichi ADC designed now. And obviously, you have more latest technology, they used it. And the payload really works it because it's chemo payload. And look at the response rates in the patients. Beautiful, actually. So this is my favorite ADC till today. There are also decisions, not necessarily efficacy-driven decision. You will have to use a safety-driven decision. This is the work I've closely witnessed at Genentech. What they have done, they use two kinds of payload. One is MMAE, other one is NMS 818. It's DNA damaging payload. This target LGR5 expressed in the normal colon stem cell, in addition to expressing the cancer. So when you express the colon stem cells, the payload gets delivered into the stem cell. The stem cells get delivered, killed, and then the villi will collapse. That happens only if the cells ADCs use the DNA damaging payload. But if you use MMAE, which is a reversible payload, they're not that drastic. So in other words, the DNA damaging payload is a reversible binding mechanism of action. MMA is a reversible binding mechanism of action. So in this case, when you have a normal expression liability in such as a critical cell population, such as stem cells, you want to stay away from the DNA damaging payload. So that is why it's important what payload you want to check. This is the paper we published a few years ago. It's the efficacy-driven decision. Here we have to use a payload which DNA damaging payload we have to use, not the reversible payload such as MMAE. In this case, cancer stem cell have expression of this target, but also the bulk tumors too. So both have the target. If you use the DNA damaging payload, both cancer stem cells as well as the bulk tumors get killed. But if you use the MMAE, only bulk tumor gets killed, but not the cancer stem cells. So therefore, for effective therapy, we have to use a payload that damages both. So in this case, DNA damaging payload, we have to use it. So let's move on to shift the gear a little bit more towards different direction. We talked about target, we talked about antibody selection, we talked about the payload selection, and now we are going to talk about how to improve the window. What do you do to improve the window? And there are multiple things you can do it. I am actually very fortunate to take on the challenging problem back in 2001, when people were struggling to make uniform homogeneous ADCs. I took on a challenge. I would want to make site-specific ADC by engineering cysteines in the antibody. Remember, there are 32 cysteines in antibodies. I went ahead and engineered a single cysteine into it, very successful. And later on, so many people went on trying doing this, so many companies came, and there were 30 site-specific ADCs are in the clinic. So Genentech was the first to make this discovery, and these were published in Nature Biotech, 2 back-to-back Nature Biotech papers, 2008 and 2012. You are most welcome to go ahead and read all of them. Why it is so special? Because making homogeneous ADC is very critical, because your molecules are going to be homogeneous and special and safer, and they also, where you attach is also equally important, because site of attachment had a huge role on it. That's the second Nature Biotech paper. I'm not going to go about too many details here, but homogeneous ADC is the critical factor. It improved the therapeutic index, it improved the safety, but efficacy was untouched in this case. So this another paper came up by MedImmune AstraZeneca 5 years ago. Beautiful paper. I personally like this paper a lot compared to any other papers past 5 years. What they have done is they actually made a tetravalent antibody. In other words, typical antibody is a bivalent. Now you increase the valency by 2-fold. And they used it, attached a payload, and tested in the cells. Beautiful internalization, lot of delivery, and then you got very good killing. So it worked beautifully, but unfortunately, it did not succeed in the clinic for various reasons. I cannot explain that today, but I know the reasons what it is. There's another paper, again, for a different company, and what they have done is they actually made a bispecific antibody attaching one arm, attaching R2, binding R2, other arm attaching CD63. When you combine these 2 antigens with a bispecific antibody, you have got a good internalization, a good efficacy. Again, another interesting idea to think about it moving forward. But I personally like this story a lot. When you inject ADCs into the system, they go all over the body. They're not going to the just tumor tissue directly. When they go all over the body, some of them leak into the body systems, tissue systems, through penocytosis, macropenocytosis. I'm sure you all heard about macropenocytosis. Every macromolecule get inside the cell through macropenocytosis after you dose certain range. ADCs are struggling with this problem because you have dealing with this toxicity. How do we deal with it? This Agenesis company, they don't exist now, and they've done a beautiful experiment actually. They actually decreased and made it positive surface to acidic surface on antibody. When they did it, the antibodies do not macropenocytosis. They decrease macropenocytosis. Something to think about it. This is out-of-box thinking, in my opinion. Then another story, this is from Seattle Genetics. No, not from Seattle, sorry. This is a different group. So they used a different linker, a glue valcid linker. Everyone uses valcid linker. Valcid linker problem is in the mouse plasma, it gets cleaved by esterases. It's not a problem in the human or monkey. Because of that, you have a poor efficacy in the mouse. So you're not really calculating good efficacy. So what these people have done, they added a glutamine next to it. They got rid of the esterase cleavage, non-specific esterase cleavage. Again, another interesting idea. I like this paper from Seattle Genetics. What they've done, they decreased the hydrophobicity of the antibody-drug conjugate by changing the linker. What they have done, they made a branch linker instead of a straightforward linker. By doing branch linker, PEG linker, it is actually protecting the linker and payload, and by doing that, it decreases the hydrophobicity. When you decrease the hydrophobicity, you are actually making the ADCs not to absorb by liver, and therefore you have longer half-life, longer efficacy, and better safety. Beautiful story again. Another Nature Biotech paper. So another concept, this is Marsana. I like it personally. What they have done, they used a hydrophilic polymer, attached the payload multiple times. 20 times they attached it. 20 attachment is a nightmare in the field, but this first time they saw it, you can attach it without compromising half-life. It works beautifully. They're trying to study them in the clinic. Let's see what the story they will have, whether it's going to work or not. These are all good, but some of them didn't make it to clinic. I like personally these stories. I wish some of this would come into clinic. I have no idea what the decision behind not bringing them into clinic. If I am the boss, I'm the decision maker, I will bring some of these actually, because they're going to make a difference. Meanwhile, so many different kind of precision medicine is coming. You know, you have one class, the function blocking antibodies such as Herceptin, Rituxan, Avastin, anti-PD-L1, which can bind the tumor cells and make it work. Some of them fantastic stories. We have approved drugs such as Adcetris, Kadcyla, 10 of them now. Good stories. And now field is moved on thinking about cortisol. Everyone talks about cortisol. Nothing special about it. Okay. That they're designed, engineered precision medicines. Nothing special about cortisol that antibodies cannot do it. We can design antibody which can do better than cortisol, actually. Yes, you can do that. And there are also antibodies you can engineer to attract the T cell to the tumor cell and kill the tumor cell. Yes, you can do that. Also now people, we can think about bispecific ADCs combining the targets. Yes, you can do that. Many things you can engineer. These are all beautiful ideas. And I want to spend last few minutes quickly on combination therapy. Everyone is going about Gaga and combination therapy. Clinician declaring success. Fantastic. When you combine the drugs, yes, it will work because they make different mechanism of action. And example, here, a beautiful story, what they have done is they treated the patient a PD-L1 before they treated with the ADC. And this is actually in the animals they have done, animal models. When they treated the PD-L1 in these mice, what happened? The expression of PD-1 and went up. When the expression of PD-1 went, now you do combination of TDM1, ADC, and CTLA4 and PD-1, fantastic activity. But just think about it. Who is going to pay for this? Each target therapy costs $100,000 per year. So do you think patient should pay $100,000 for TDM1, patient should pay for $100,000 PD-1 or CTLA4? This is just impossible. Yes, it's going to work. You're going to break the financial healthcare system completely. I do not want to have combination therapies. I want to have one molecule can kill the entire tumor. Why should I pay for 3 drugs in combo? Tell me. As a patient, I do not want to do that. As a scientist, I don't want my patients to do that. So this is what scientists should think. Should think a molecule which can answer all these questions. Think about a molecule combining the 3-4 drugs. Think about a molecule doesn't fail in the clinic. Why should 10 out of 10, 90 only work? Why not 90 out of 100 should work? Why not? Are we not designing properly these molecules? So this is a challenge for my fellow scientists. Can we do this better job? I'm sure we can. We are in 21st century. There is fantastic technologies around. And if ever I want to start a company, that's what I want to do. I want to have a molecule which combines all these 3-4. I want to have a molecule to be successful. 50 out of 100, I want my molecules to be successful. Otherwise, I will not start the company. I do not want to do that. Poor research. Thank you for listening my story. To all my passionate ADC colleagues in the pharma biotech and all these wonderful creators sacrifice their life, and so many patients enrolled into the ADC clinical trials. 90% of them have failed. Let's make their hopes alive. Thank you. Hi.
Sorry about that, Dr. Jagath Reddy. They just had to get me in. Yeah. So I could see the passion in your presentation. You got a little emotional too, and that's fair because that tells you how much you care. And I think you ended it well by appealing to all your passionate colleagues, and that molecule will be found. We wish you. Right now, there are no questions for you. I'm sure this presentation of yours would have generated quite a few questions. And if there are any, Ashish will send them to you offline. Thank you so much. We wish you the very best in your work, and we wish Science Gurus the very best going forward too. Thank you.